next, we'll be welcoming Beatrice Sim. She'll be presenting on novel fungal diagnostics, and she's one of our online cohort today. So, Dr. Beatrice Sim is an infectious diseases physician who's trained in Queensland and South Australia before undertaking an immunocompromised host infectious diseases fellowship at the Peter MacCallum in Melbourne and a transplant ID fellowship at Duke University in North Carolina. She's currently completing a PhD in novel viral diagnostic strategies through the University of Melbourne and the National Centre of Infections in Cancer.
Thank you for the invitation to speak on one of my favorite topics, novel fungal diagnostics. I'm sorry I can't be there in person today. So this is an area that is in great need of development, as highlighted by the WHO this year. And so in this talk, I plan to give you a bit of an overview of the progress and potential future diagnostic tools at various quite early stages of development. So there are four broad categories of tests. So culture-based, direct visualization, antigen testing, and molecular testing. And the problem with all of these is none of them are perfect. Serology is also available, but more in use for North American endemic fungi, so I will not really discuss this today. For this audience, I won't go into the minutiae of the differences between these tests, but suffice to say that there is a bit of a balance between the need to obtain invasive samples and the ability to get granular detail about species and susceptibility testing. As Daniel mentioned, MSG and ERTC criteria tried to summarize all of these diagnostic methods and also take into account the host and their clinical picture, but this is more designed for research rather than clinical diagnosis and therefore has its own problems. The newer pathogen detection tools are primarily molecular-based tools, and in routine clinical use, we're primarily familiar with PCR. The issue with PCR for diagnostics is that generally you have to know what you're looking for. So, for example, you have to suspect Aspergillus to be able to send out an Aspergillus PCR or Mucorpha, Mucorales PCR, and so on and so forth. Sanger sequencing kind of addresses that a little bit because you're able to send for a broad range PCR. For example, looking for, the 18S ribosome, if it's, as in this case, we're talking about fungal infections. But then the category of tests that I want to talk about is the new and emerging category of pathogen detection molecular tests, broadly referred to as next generation sequencing. The main difference, I think, between Sanger sequencing and NGS is the sheer volume at which you can perform this. So instead of sequencing a single DNA fragment at a time, as in Sanger sequencing, next-gen sequences millions of fragments simultaneously per run, which means that it can sequence hundreds to thousands of genes at a time, or in this case, hundreds to thousands of pathogens. All of these platforms, in theory, can be performed on every different type of body fluid or tissue, but because of the relative fragility of our transplant patients, the ideal test would be something that can be done non-invasively, by which I mean as a blood test. So not only would you be able to avoid invasive procedures, you can also potentially collect serial samples to monitor things like response to treatment, relapse, and potentially predict complications if, for example, you can show that a higher burden of organism corresponds to increased complications. However, there are still some problems that need to be worked out. And namely, we don't know exactly who to use these tests in, and the use of these tests is not clearly defined. We don't know all that much about its sensitivity or specificity in the real-world clinical use. And once again, with most of this molecular tests at the moment, the inability to do susceptibility testing still poses a bit of an issue. So at least there have been at least three non-invasive next-gen sequencing tests that have investigated for invasive fungal infection. That's a plasma Aspergillus PCR that was developed at Stanford that one of my co-fellows at Duke, Jordan Ma, has been working on. The serum Mucorales PCR that was developed by the French mycosis group. And finally, the commercially available plasma microbial cell-free DNA sequencing test, bit of a mouthful, so known colloquially as the carriers test. And that was developed by the eponymous carriers group. And the latter is what I'll focus on in the interest of time, and also because it's what I've been working on here too. But I have included at the bottom there the reference to the first two studies for your information. So the carriers test is a single commercially available blood test that theoretically allows pathogen agnostic detection of approximately 400 fungi and many other bacterial and viruses, and all from a single non-invasive test. The company in the US spooks about a 48-hour turnaround, which relative to normal culture-based methods, it's insanely quick. The utility of the test has been shown in other scenarios, so non-IFI scenarios, so things like culture-negative infective endocarditis, but certainly there is less evidence in the immunocompromised host or in invasive fungal infection. So what evidence do we have? So this is a study done by the group at the Fred Hutch in Seattle. They looked at stored plasma samples for stem cell transplant recipients with invasive fungal pneumonia only. And they found that of the 75 patients with proven or probable invasive fungal infection per MSG criteria, the Caris test detected at least one pathogenic mold in 38 patients, so a sensitivity of about 51%. When they limited the samples to those collected within 3 days of diagnosis, this improved to 61%. However, it's worthwhile noting that in the Aspergillus group, this actually didn't perform better than galactomannan, but it did do better than PCR and culture. The other thing to also note is that sensitivity looked like it was higher for non-Aspergillus invasive fungal infection at 79%, which is perhaps promising because we don't really have a lot of good diagnostics for a lot of these fungal infections in routine clinical use. Of note, there were no molds detected in any of their control patients, so the patients without IFI, so giving that 100% specificity. And the other thing that was important was that prior antifungal therapy did not affect the results either, which can sometimes be a problem with some of the other fungal markers. They also looked at the timing of sample collection and essentially found that the concentration of DNA detected peaked at the time of clinical diagnosis. So by the time you were suspicious enough to collect your bronchoscopy or your biopsy or whatever you're going to use to make a diagnosis, you were pretty likely to have a positive test. The earliest positivity was about 3 weeks prior to the MSG diagnosis. And in this small study, there was no clear relationship between, chest imaging findings or time on antifungals, or unfortunately, either the concentration of DNA detected, but that certainly would be another area of interest, that ability to monitor response to treatment from a single blood test. So data in the solid organ transplant groups even thinner on the ground. And so to address this, we published our results from our case control study in CID that we'd done earlier this year. So this was done using prospectively collected archived plasma samples from the Aspergillus Technology Consortium, or the ASTEC repository. In brief, this included 65 patients with proven or probable invasive fungal infection who had been undergoing prospective surveillance, and 65 matched controls without IFI. Patients included a mixed real-world cohort of immunocompromised patients, so those with stem cell transplants, solid organ transplants, as well as those receiving chemotherapy for hematological malignancies. We found very similar results to the Fred Hutch study, so sensitivity still wasn't fantastic, and maybe that's because we haven't quite worked out who to do it in or maybe when to do this test. But similarly, none of our controls had any pathogen detection, making that specificity 100% still. Interestingly, for our study, the timing of specimen collection didn't particularly affect our yield. Proven IFD similarly had a higher sensitivity than probable, and that once again is perhaps reflecting that imperfect MSG gold standard definition that Daniel alluded to earlier. We also noted that the non-Aspergillus species, in our case Mucorales, had a higher sensitivity as well than Aspergillus species. Even though our numbers were small, we also did some logistic regression analyses to look at the impact of other host factors, especially those ones that have previously been found to impact our other fungal biomarkers. And we found that the odds of identifying a causative pathogen was not altered by things like active antifungal therapy, location of the disease, so lung versus disseminated, and patients who are neutropenic versus those who were not. And all of these have previously been identified as factors that can influence other biomarkers like galactomannan and beta-D-glucan. So promising, but definitely still work to go, especially in the absence of other better fungal markers. So that's pathogen detection. What about using host response as a diagnostic? So one suggested method is using transcriptomics to try to capture a pathogen-specific host response. So trying to see if there's a signature that we can use in gene expression that corresponds to a specific pathogen. This has been used to differentiate between infected and non-infected host, and those with bacterial versus viral pathogens, but fungal diagnostics, as per usual, is not very well investigated. And the further complicating factor in transplant patients is that any transcriptomic signature is also very likely to be affected by your host immunosuppression as well. And this can be seen in this study, which is a murine model of invasive aspergillus done by one of my colleagues here at Duke, Julie Steinbrink. In this study, they looked at mice with varying levels of immunosuppression. There were 3 groups, so no immunosuppression, cyclophosphamide, and mice on steroids. In the non-immunosuppressed group, they found quite significantly different, differentially expressed genes, about 2,718 genes to be precise. And they used, through bioinformatics, they used that difference to generate a transcriptomic signature to distinguish between mice with aspergillus versus mice without aspergillus. And as you can see in this graph on the right, hopefully you can see my mouse, this actually held up pretty well in this blue non-infected mice. So you can see that using that signature, there was quite a difference between those that were infected versus those who are not. Whereas on the other hand, it held up, unfortunately, less well, I'm sure that's a word, in the cyclophosphamide group, in the green here, and even worse in the steroid group. So to account for that, they looked at they accounted for the impact of immunosuppression by using a set of normalized, they accounted for the impact of immunosuppression on the normalized gene expression values by using a prime model of medication gene expression, the effect of medication on gene expression. And they ran the analysis again to look for a classifier. And in the final gene set, they included 169 genes. And you can see that the new classifier actually distinguished infected mice in the sort of the darker colors here. So once again, the blue being the ones with no immunosuppression, green cyclophosphamide, red steroids, and the lighter color are the ones that were infected versus the darker colors, which were not. And you can see that that actually distinguished it very well. And the idea is that this can be applied to other fungal infections, so ones that have poorer diagnostics. At the moment, I'm working on a study looking at gene expression in Cryptococcus as compared to those in bacterial, viral, or no infections in a set of real-world patients. So certainly some practical applications there, potentially. Right, radiological techniques have also always been of interest. The advent of FDG PET-CT has proven useful in both detection and monitoring of invasive fungal infection. And this is clearly illustrated in the scans on the right from a study by Abby Douglas, who spearheads quite a lot of this work in Australia. So in the first paired scans, you can see that the PET-CT lights up where the plain CT doesn't, aiding in the diagnosis of, I think this one was disseminated candidiasis. In the second panel, you can see that while the pulmonary lesion looks the same, once again, I hope you see my mouse, on the CT scan between the interval scans, the PET avidity has actually gone down in that same lesion after a course of treatment. So definitely by itself, PET-CT has proven a useful tool in both diagnostics and also monitoring response to treatment, but it could be made more useful by improving its pathogen specificity. And this is the subject of a clinical trial run that we ran at Peter Mac, or running at the Peter Mac with Abby. So the way we have approached improving pathogen specificity of the scans is by harnessing a molecule called siderophore. So siderophore, not drawn to scale, are molecules produced by microbes to chelate iron that they need for growth, especially during infection. The reason why they're good targets for use is because they're quite specific to different microbes. So what happens in this extremely high-tech, non-AI generated animation is that when the fungus releases the siderophore, it goes out to chelate iron, it finds the iron, and then when it finds the iron, it can be reuptaken into the organism by siderophore iron transporters, which are also very specific to the organism. So what that means is, theoretically, if you radiolabel that siderophore, administer it to the patient, then the aspergillus inside the patient should be able to take it up, and you can localize the infection by PET scanning it. And if you give a siderophore that's specific to a particular organism and the PET scan lights up at that place, well, that can give you a diagnosis of that organism. So Aspergillus fumigatus, for example. And that has been our focus. In Aspergillus fumigatus specifically, there are two different siderophores that are produced that could be potentially targets for radiolabeling, TAF-C and fusarenine C. The one that we picked was TAF-C. It's been shown to be an ideal target because it's naturally occurring, secreted by Aspergillus fumigatus, and it's already been found in the urine of infected patients, so we already know that it's there. It's also taken up by a siderophore iron transporter that is unique to the organism and also highly upregulated during infection. And then, of course, the important point is that it's not taken up by a human system, so it's unlikely to have adverse effects. So animal models have already been done. In the graph on the left, you can see in the black bars there, that's gallium labeled TAF-C, and that amount detected in this infected mouse is corresponds to the severity of infection. So more detected, the more severe it is, and practically nothing there in the control. The scan in the middle shows the actual uptake, so that sort of dark color in the lungs there, in the mice between severe, mild, and control infection. And the scan on the right shows a PET scan, looking at the difference between uptake in infected versus uninfected mice, where the infection can be seen in that green-yellow. So therefore, we're currently conducting a pilot first-in-human study that we've termed the specific study. This is a proof of concept study where we are recruiting 10 patients with known, proven, or probable aspergillus within 2 weeks of their original diagnosis. We then administer to the patient the gallium-labeled TEFC and then PET scan them at 15, 60, and 180 minutes to see how that lights up and then washes out. The primary objective is to demonstrate specific uptake of the gallium-labeled TAF-C radiotracer in the areas of known, proven, and probable invasive aspergillus infection. And of course, the secondary objectives are that of safety. So here are the results from our first patient that was presented at ID Week last year. So this was a patient with probable Aspergillus fumigatus in the setting of AML, and the diagnosis of invasive Aspergillus was made on basis of a positive Aspergillus galactomannan and a PCR on BAL. Patient was already on voriconazole for about 10 days when the PET scan was performed. As you can see, and the scans here, and I'll show you a bit more detail a bit closer up later. There was focal uptake in multiple pulmonary lesions that we've highlighted in the green there, and that was at SUV max of 3.5. And you can sort of see, or you will sort of see, that there was synchronous washout over time. There was early renal excretion in this patient and no adverse effects. So really very exciting stuff. So here's a closer look at the scan, and hopefully you can see it rotating there. Once again, the areas of known infection are sort of the same-ish color as the heart, so about that same blood pool level. And we think that potentially that might have been because of the duration of time that the patient had already been on voriconazole. We've highlighted those lesions there, and you can see as the time sort of went by, the tracer washed out. And that's the sort of cross-sectional scans as where it corresponded to. So really exciting stuff. I'm looking forward to getting back to Australia and see how many more patients we can recruit. So that's being run by Abby Douglas at the Peter Mac. So all in all, that's a bit of a quick summary, but a very exciting time, I think, with the new developments all in the works.
That's wonderful, Beatrice. Thank you. And are you going to be able to stay online for questions after as part of the Q&A?
Yeah, I'll be around.
Wonderful. Excellent. Okay. Well,